Imagine like a scientific conversation happening across decades.
Yeah, between two key figures who never really, collaborated directly in the lab, but their ideas definitely did.
Exactly. Two brilliant minds working decades apart, different organisms even, but they basically laid the foundation for modern genetics.
It's not just history, though, is it? It feels more like a detective story.
It does, where the clues only really snap together later on.
Precisely. So today, we're taking a deep dive into, well, the origin story of mutation. That word, that concept.
And how absolutely pivotal it became for understanding heredity. It's a journey that really did reshape biology.
Totally. Our mission, if you will, is to unpack this sort of implicit dialogue between Hugo de Vries,
The guy who actually coined the term mutation, right?
that's him, and Thomas Hunt Morgan, whose, let's face it, groundbreaking work with fruit flies really cemented how genes work.
Well, you're about to hear how this fundamental idea, sudden heritable changes, just completely transform biology.
It led to Nobel Prizes, discoveries that underpin molecular genetics even now. We'll dig into the messy scientific world before them.
Right. What was the thinking back then?
And these dramatic sports, these odd plants de Vries found.
The evening primroses.
Yep. And then the big one, Morgan finding that single white-eyed fruit fly.
Ah, the famous fly. And how that discovery, plus others, led them to actually mapping genes on chromosomes, physically locating them.
It's an amazing story. So yeah, what does it all mean for understanding life? Let's unpack this.
Okay, so set the scene for us. Turn to the 20th century. Heredity, what did it look like?
Honestly, it was foggy, speculative. Darwin's theory of evolution was huge, obviously, but it had this gap.
How variation actually happened and how it
Uh,
got passed on.
Exactly. That was the missing piece. The main idea floating around was often this blending inheritance.
Like mixing paint. Offspring are just an average of the parents.
Right, which, if you think about it, doesn't explain how traits can skip a generation and reappear, or why variation doesn't just get diluted away.
So the very fuel for natural selection variation was this big question mark.
Huge. And that's the intellectual space de Vries was working in. He saw this gap.
And he looked at evening primroses. Onothera lamarckiana. Why those?
Well, he started noticing these really striking, sudden variations popping up, plants that looked radically different from the parents.
He called them sports, right?
He did. And crucially, they bred true. They passed these new traits on. It looked like new species were just appearing.
Wow. I mean, imagine seeing that when the prevailing view was slow, gradual change.
Absolutely. And right around the same time, 1900-ish, Mendel's work was rediscovered, those discrete units of inheritance.
Providing mathematical framework, but still, where were these genes physically?
Nobody really knew, so you had this tension. Mendel's neat packets of inheritance, Darwin's gradualism, and now de Vries seeing these big jumps.
And were there scientists, the biometricians, who were still focused on continuous, measurable variation in populations?
Oh yes, Carl Pearson and his group. They emphasized gradual changes across populations and struggled to fit Mendel's discrete factors and de Vries's jumps into their models. It was a real battle of ideas.
Okay, so into this fray steps de Vries with his big idea.
Right. Between 1901 and 1903, he publishes his mutations theory.
That new species could arise not just slowly, but through these large, sudden shifts. Mutation.
Exactly. It wasn't just giving it a name. It was a fundamentally different way of thinking about evolutionary change. Maybe it wasn't always a crawl.
That's a pretty radical proposal for the time.
Definitely. And he backed it up. He spent, 13 years breeding Onothera, meticulously documenting these changes, big and small.
He called the stable new forms elementary species.
Yes. His work, even if the specifics were later debated, provided the theoretical push. It made people look for discrete, sudden changes.
And that's the direct link to Thomas Hunt Morgan.
It absolutely is.
Yeah.
Morgan, working with the Drosophila melanogaster, the common fruit fly.
Much easier to breed than primroses, I imagine.
Oh, much faster life cycle, easier to keep large numbers, perfect for experimental work, and he was explicitly looking for mutations inspired by de Vries.
Okay, so he's breeding thousands upon thousands of these flies.
Normal red-eyed flies.
Yeah.
And then, in 1910, bam.
The white-eyed male. Amidst all the red eyes.
Huh.
What did that single fly mean in that moment?
Everything. It wasn't just a mutation, it was the first clear-cut sex-linked mutation ever documented.
Meaning its inheritance pattern followed the sex chromosome.
Precisely. It was clearly linked to the X chromosome. This was massive because it tied a specific heritable trait, eye color, not just to an abstract gene, but to a concrete physical structure, a specific chromosome.
It answered the question, where do genes live?
Or started to.
It started to.
Yeah.
It put genes on the map, literally. So that white-eyed fly wasn't just a discovery, it was like a key turning in the lock.
That's a great way to put it, yeah.
So once that door creaked open, what happened next in Morgan's lab? The famous fly room.
Well, it kicked off an explosion of discovery. Morgan and his brilliant students, Sturtevant, Bridges, Muller, They started finding more mutations.
And crucially, they started noticing that some traits seemed to be inherited together more often than expected. They didn't always assort independently like Mendel's peas.
Exactly, they were linked, which strongly suggested that the genes for these traits weren't just floating around on the chromosome, they were sitting on it in a specific linear order, like beads on a string. And based on that, Sturtevant drew the very first genetic linkage map, literally mapping the relative positions of genes on a chromosome.
That's just incredible. From a theoretical idea to a physical map, a blueprint of heredity.
It really was the birth of quantitative genetics in many ways. They weren't just observing, they were measuring and mapping.
Okay, let's circle back to de Vries for a second.
Yeah.
How did his work and Morgan's ultimately connect or reconcile?
Well, it's interesting. De Vries himself acknowledged Morgan's work later on, and it turned out that many of those dramatic mutations de Vries saw in his evening primroses
Weren't quite what he thought.
they weren't simple gene mutations like Morgan's white eye. They were actually more complex chromosomal rearrangements, duplications, deletions. Even if the specifics of his unathera examples were reinterpreted, his core concept, the idea of sudden, discrete, heritable change as a driving force, was revolutionary.
It gave people the framework, the thing to look for.
Exactly. It primed the pump. It gave Morgan and others the theoretical impetus to look for mutants in a systematic, experimental way. Morgan's work then refined the concept.
Showing that, yes, these sudden changes happen, and they can be changes in single genes, located at specific spots on chromosomes.
Right. It showed the finer scale. So you have a spectrum from single gene changes like white eyes to larger chromosomal changes like those in Enothera, all falling under this broad umbrella of mutation that de Vries introduced.
It's fascinating how they almost needed each other, even though they worked so differently. What was the most critical piece of de Vries's legacy for Morgan, do you think?
I think it really was the term mutation itself and the idea of discrete novelty arising suddenly. It sparked that systematic search.
It shifted genetics towards being an experimental science.
Absolutely. Find the mutants, cross them, figure out the inheritance patterns, map the genes. That became the paradigm, driven by Morgan's lab using Drosophila. And de Vries saw that Morgan's mapping work completed the picture he had started.
Discrete particles of inheritance, physically located, rearranging.
Yeah, the foundation for molecular genetics was truly laid right there.
So it's less about who was right and more about how the bold theory needed the rigorous experiment.
Precisely. The theory provided the target, the experiment provided the proof and the mechanism.
And connecting this to the bigger evolutionary picture, de Vries was seen as a saltationist, right? Believing in big jumps in evolution. How did that fit with Darwin?
Well, initially it seemed quite opposed to Darwin's gradualism, but Morgan's work, firmly rooted in Mendelism and chromosomes, provided a way to bridge that gap.
How so?
Because mutations, whether small gene changes or larger rearrangements, provided the raw material, the discrete variations, that natural selection could then act upon. It helped form the basis of the modern synthesis of evolution later on, which brought together genetics, Darwinism, and other fields. It wasn't strictly gradualism or saltationism, it was a mix.
Okay, so wrapping this up, why does this sort of century-spanning scientific dialogue matter today? What's the takeaway?
Well, for de Vries, his legacy is introducing mutation as this central, experimentally tractable concept, moving beyond just philosophical ideas about species origins.
He made variations something you could potentially find and study in the lab.
Right. And for Morgan, de Vries's theory was the direct prompt for his empirical work. He delivered the mechanism.
Locating genes, demonstrating recombination, creating those maps. The stuff that won him the Nobel Prize in 1933.
Exactly. And that established Drosophila as the model organism and transformed genetics into this powerful, quantitative, model-driven science we know.
What strikes me is how a visionary idea, even if its first examples turn out to be, let's say, not quite the perfect illustration, like de Vries's primroses being chromosomal things.
Right, rather than simple gene changes.
And that idea can still be the critical spark needed for the empirical work that does hit the nail on the head and changes everything.
Absolutely. We've traced how de Vries's bold theory, the mutations theory, gave us the concept of sudden, discrete variation. That was the spark.
And that spark ignited Morgan's incredibly rigorous experiments with the fruit fly.
Which gave us the physical reality. Genes lined up on chromosomes capable of recombining.
It's this amazing interplay. Theory informs experiment. Experiment refines theory. That's how genetics leaped from speculation to a real quantitative science.
Yeah, it really is. And consider how often scientific progress works like that. A bold idea, maybe imperfect at first, provides the crucial push for the experiments that solidify our understanding. Which makes you wonder, doesn't it?
What other bold, maybe slightly off ideas are out there right now?
Exactly. Waiting for the right experimentalist, the right system to take them from concept to concrete reality and, reshape our world again.
It's a privilege to explore this kind of scientific conversation with you, showing how progress happens often across generations and different ways of thinking.